Hello friends, today we will gain knowledge about transcription regulation in eukaryotes, and our main objectives will be: number first, transcription regulation in eukaryotes; number second, conserved mechanism of regulation; number third, eukaryotic activators; number four, signal integration; number five, combinatorial control of transcription; number six, transcriptional repressors and control of transcriptional regulator; and the last one will be gene silencing. So first and foremost, a general introduction about the transcriptional regulation in eukaryotes. Transcriptional regulation is the means by which a cell regulates the conversion of DNA to RNA transcription, thereby orchestrating gene activity. A single gene can be regulated in a range of ways, from altering the number of copies of RNA that are transcribed to the temporal control of when the gene is transcribed. This control allows the cell or organism to respond to a variety of intra- and extracellular signals and thus mount a response. The regulation of transcription is a vital process in all living organisms. It is orchestrated by transcription factors and other proteins working in concert to finely tune the amount of RNA being produced through a variety of mechanisms. There are a number of mechanisms through which eukaryotic polymerase activity can be controlled. These mechanisms are conserved and can be generally grouped into three main areas. Number first, control over polymerase access to the gene. This is the broadest of the three control mechanisms. This includes the functions of histone remodeling enzymes, transcription factors, enhancers and repressors, and many other complexes. Number second, productive elongation of the RNA transcript. Once polymerase is bound to a promoter, it requires another set of factors to allow it to escape the promoter complex and begin successfully transcribing RNA. And third one, termination of the polymerase. A number of factors which have been found to control how and when termination occurs, which will dictate the fate of RNA transcript. All three of these systems work in concert to integrate signals from the cell and change the transcriptional program accordingly. In prokaryotic systems, the basal transcription state is a non-restrictive, but eukaryotes have a restrictive basal state, which requires the requirement of other factors in order to generate RNA transcripts. This difference is largely due to the compaction of the eukaryotic genome by winding DNA around histones to form higher-order structures. A large part of gene regulation occurs through transcription factors that either recruit or inhibit the binding of the general transcription machinery and/or the polymerase. Transcription factors. First, they regulate gene expression by helping or inhibiting transcription initiation, for example, by helping to recruit RNA polymerase. Number second, they bind to DNA in a sequence-specific manner. And number third, they play an important part in the much larger picture of expression regulation. We have already outlined the three general mechanisms of transcriptional regulation. Now let's go through each of them in detail. Regulation at the level of chromatin state. In eukaryotes, genomic DNA is highly compacted via histones in order to be able to fit into the nucleus. Significant portions of eukaryotic genome are silenced through histone modifications and thus are inaccessible to the polymerase or their cofactors. Histone rearrangement is facilitated by post-translational modifications to the tails of the core histones. A wide variety of modifications such as addition or removal of methyl groups, acetyl groups, phosphates, and ubiquitin can be made by enzymes such as the histone acetyltransferases (HATs), histone methyltransferases (HMTs), and histone deacetylases (HDACs), among others. Histone modifications serve to recruit other proteins which can either increase the compaction of the chromatin and sequester promoter elements or to increase the spacing between histones and allow the association of transcription factors or polymerase on open DNA. Number second, regulation through transcription factors and enhancers. First, transcription factors. Transcription factors are proteins that bind to specific DNA sequences in order to regulate the expression of a given gene. The power of transcription factors resides in their ability to activate and/or repress wide repertoires of downstream target genes. Transcription factors function through a wide variety of mechanisms. Often they are at the end of signal transduction pathway that functions to change something about the factor, like its subcellular localization or its activity. Post-translation modifications to transcription factors located in the cytosol can cause them to translocate to the nucleus, where they can interact with their corresponding enhancers. Others are already in the nucleus and are modified to enable the interaction with partner transcription factors. Transcription factors may be divided into two main categories: activators and repressors. While activators can interact directly or indirectly with the core machinery of transcription through enhancer binding, repressors predominantly recruit co-repressor complexes, leading to transcriptional repression by chromatin condensation of enhancer regions. Tight regulatory control is achieved by the highly dynamic nature of transcription factors. Again, many different mechanisms exist to control whether a transcription factor is active. These mechanisms include control over protein, localization or control over whether protein can bind DNA. The second one is regulation through enhancers. Enhancers or cis-regulatory modules or elements CRM or CRE are non-coding DNA sequences containing multiple activator and repressor binding sites. The hallmark of enhancers is that, unlike promoter elements, they can be downstream, upstream, or within an intron and can function in either orientation relative to the promoter. Each enhancer is responsible for a subset of the total gene expression pattern. Enhancers increase gene promoter activity either in all tissues or in a regulated manner. Enhancers can be either proximal, 5' upstream to the promoter, or within the first intron of the regulated gene, or distal in introns of neighboring genes or entegenic regions far away from the locus. Through DNA looping, active enhancers contact the promoter dependently of the DNA binding motif promoter specificity. Promoter-enhancer dichotomy provides the basis for the functional interaction between transcription factors and transcriptional core machinery to trigger RNA polymerase second escape from the promoter. Now let's come to our second objective, transcriptional activators. Eukaryotic transcriptional activators are modular proteins that are typically composed of a sequence-specific DNA binding domain and activating region. It's now generally accepted that activators interact through their activating regions with components of the RNA polymerase second transcriptional apparatus that bind promoters. According to their amino acid constitution, activation domains have been classified as acidic, for example, the viral VP16 and the yeast GAL4, glutamine-rich, for example, SV1, and the proline-rich, for example, the CTF. Binding of the activator to DNA serves merely to locate it to a position from which activation domain can activate transcription. Activators have a regulatory role and are synthesized or activated at specific times or in specific tissues. These factors are therefore responsible for the control of transcription patterns in space and time. The sequences that they bind are called response elements. Response elements may be located in promoters or in enhancers. Binding of an activator to the response element is required to allow RNA polymerase to initiate transcription. The difference from constitutively active activators is that the protein is either available or is active only under certain conditions, which determine when gene is to be expressed. Many genes, especially in higher eukaryotes, are activated not just by one, but by several activators that act in concert. Dear friends, activator has separate domains that bind DNA and activate transcription. Each domain behaves as a separate module that functions independently when it's linked to a domain to the other type. Activators that are more or less ubiquitous are assumed to be available to any promoter that has a copy of the element that they recognize. Common elements recognized by ubiquitous activators include the CAAT box, GC box, and the octamer. An activator typically has consensus sequence less than 10 base pair, but actually covers a length of 20 base pairs of DNA. Usually a particular consensus sequence is recognized by corresponding activator or by member of a family of factors. However, sometimes a particular promoter sequence can be recognized by one of several activators. Dear friends, our third objective, that's conserved mechanism of transcriptional regulation. Crucial remodeling of the chromatin takes place in stages. Some DNA binding trans activators have significant affinity for their binding sites, even when the sites are within condensed chromatin. Binding of one trans activator may facilitate the binding of others, gradually displacing some nucleosomes. The bound trans activators can then interact directly with HATs or enzyme complexes such as SWI or SNF or both, accelerating the remodeling of the surrounding chromatin. In this way, a bound trans activator can draw in other components necessary for further chromatin remodeling to permit transcription of specific genes. The bound trans activator, generally acting through complexes such as TF2D or mediator, and greatly facilitate formation of the pre-initiation transcription complex. Complexity in these regulatory circuits is the rule rather than the exception, with multiple DNA-bound trans activators promoting transcription. The script can change from one promoter to another, but most promoters seem to require a precisely ordered assembly of components to initiate transcription. The assembly process is not always fast. At some genes, it may take minutes. At certain genes in higher eukaryotes, the process can take days. Now let's deliberate on our fourth objective, that is signal integration. Transcriptional initiation, termination, and regulation are mediated by DNA looping, which brings together promoters, enhancers, transcription factors, and RNA processing factors to accurately regulate gene expression. The configuration of the genome is essential for enhancer-promoter proximity. Cell fate decisions are mediated upon highly dynamic genomic reorganization at interface to modularly switch on or off entire gene regulatory networks through short to long-range chromatin rearrangements. Related studies demonstrate that eukaryotic genomes are partitioned in structural and functional units around a megabase called topological association domain (TADS), containing dozens of genes regulated by hundreds of enhancers distributed within large genomic regions containing only non-coding sequences. The function of TADS is to regroup enhancers and promoters interacting together within a single large functional domain instead of having them spread in different TADS. TAD boundaries are often composed by housekeeping genes, tRNAs, other highly expressed sequences, and short interspersed elements SINE. While these genes may take advantage of their border position to be ubiquitously expressed, they are not directly linked with TAD edge formation. The specific molecules identified at boundaries of TADS are called insulators or architectural proteins because they not only block enhancer leaky expression but also ensure an accurate compartmentalization of cis regulatory inputs to the targeted promoter. These insulators are DNA-binding proteins like CTCF and TF-ters that help recruiting structural partners such as cohesins and condensins. The localization and binding of the architectural proteins to their corresponding binding sites is regulated by post-translational modifications. High occupancy sites are usually conserved and static, where intra-TADS sites are dynamic according to the state of the cell. Therefore, TADS themselves are compartmentalized in subdomains that can be called sub-TADS. Mediator proteins are the architectural proteins cooperate with cohesin. For sub-TADS larger than 100 kb and TAD boundaries, CTCF is the typical insulator found to interact with cohesin. After this, let's talk about our fifth objective, that is combinatorial control of gene expression. Combinatorial control of gene expression is the orchestration of various biological processes with high fidelity, requires a precise control over temporal and spatial expression of genes. The regulation of gene expression in eukaryotes can occur through synchronization of various steps, namely transcription, mRNA splicing, translation, and post-translational modifications. Initiation is marked by the assembly of the pre-initiation complex at the promoter of the corresponding gene. The pre-initiation complex comprised of RNA polymerase II and its auxiliary components TF2A, TF second B, TF second D, TF second E, TF second F and TF second H that bound to the core promoter can only initiate basal levels of transcription. Considering the diversity of molecular signals that the transcriptional machinery has to interpret, it would be impractical for each regulator to have a unique target. On the other hand, the specificity of signals cannot be compromised. Since each gene is under the control of more than one type and the number of cis regulatory elements, a combinatorial regulation enables the organism to have innumerable expression patterns even with a limited number of transcription factors. Now coming to our sixth objective, repressors and transcriptional repressors and control of transcriptional regulator. Repressor is a DNA or RNA binding protein that inhibits the expression of one or more genes by binding to the operator or associated silencers. A DNA binding repressor blocks the attachment of RNA polymerase to the promoter, thus preventing transcription of the genes into messenger RNA. An RNA binding repressor binds to the mRNA and prevents translation of the mRNA into protein. This blocking of expression is called repression. Eukaryotic repressors have two functional domains, a DNA binding domain and a repression domain. Variety of amino acid sequences can function as repression domains. They are relatively short, 20 amino acids, and contain high proportion of hydrophobic residues. Repression of transcription in eukaryotes is generally accomplished at the level of influencing chromatin structure. Now coming to our last objective, that is gene silencing. As the name implies, gene silencing is a technique that aims to reduce or eliminate the production of a protein from its corresponding gene, or it refers to the ability of a cell to prevent the expression of certain gene. Copy of the information encoded in a gene is made in the form of messenger RNA, mRNA. Gene silencing can occur during either transcription or translation. Imagine the gene silencing molecule as a sensor and mRNA as messages from genes that are broadcast into proteins. The molecule will censor out a specific mRNA message, preventing the corresponding protein from being broadcast into the cell, and thus silencing the gene that is providing these instructions. There are various gene silencing methods, but two of the leading and most understood methods of gene silencing are RNA interference (RNAi) and antisense RNA (AsRNA). RNA interference is a biological process in which RNA molecules inhibit gene expression, typically by causing the destruction of specific mRNA molecules. The process to silence genes first begins with the entrance of a double-stranded RNA molecule into the cell, which triggers the RNA interference pathway. The double-stranded molecule is then cut into small double-stranded fragments by an enzyme called dicer. These small fragments, which include small interfering RNAs, siRNA, and microRNAs, that's miRNA, are approximately 21 to 23 nucleotides in length. The fragments integrate into a multi-subunit protein called the RNA-induced silencing complex, RISC or RISC. One strand of the molecule called the guide strand binds to the RISC, while the other strand, known as the passenger strand, is degraded. The guide or antisense strand of the fragment that remains bound to RISC directs the sequence-specific silencing of the target mRNA molecule. The RNA interference is thought to have evolved as a cellular defense mechanism against invaders such as RNA viruses or to combat the proliferation of transposons within a cell's DNA. Now coming to antisense RNA. Antisense RNA is a single-stranded RNA that is complementary to the messenger RNA or mRNA strand, transcribed within a cell. Antisense RNA may be introduced into a cell to inhibit translation of complementary mRNA by base pairing to it and physically obstructing the translation machinery. This effect is therefore stoichiometric. The antisense oligonucleotides can affect gene expression in two ways: by using RNA's H-dependent mechanism or by using a steric blocking mechanism. RNase H-dependent oligonucleotides cause the target mRNA molecule to be degraded, while steric blocker oligonucleotides prevent translation of the mRNA molecule. Dear friends, this brings us to the end of this lecture on transcription regulation in eukaryotes. Hope you have enjoyed this lecture. Have a nice day.